In this first video, we are going to talk about collecting data to address a research question and talking about the importance of 5 increments. So we're going to be looking at the 5 increment piece, and we're going to be looking at the importance of 5 trials. Doing this experiment, we're going to be using a virtual lab, a simulation, which is the effect of light intensity. Here's our light on the rate of photosynthesis in a type of plant called Alodia. So in this virtual lab, you can control the intensity of light by either making it closer to the plant, more light intensity, or moving it further away from the plant, less light intensity. So that's what we're going to be doing. But before we do that, let's talk real quick about photosynthesis. So photosynthesis, for it to work, you need a couple ingredients on the left-hand side of our equation to make it work. So we just need carbon dioxide gas. Now, this is an underwater plant, right? Here we have it in a test tube. So Alodia is a plant that grows underwater, but there's CO2 gas dissolved within the water. So we need carbon dioxide and we need water. And from that, if the conditions are right, we get sugar in the form of glucose, and we get a waste product, oxygen. So one of the things that affects the rate of photosynthesis is definitely how much carbon dioxide there is, and also how much water there is. But we also need light. So light actually comes in and breaks water, and when it does that, it supplies the ATP, the energy, that helps us build sugar. So that's how photosynthesis works. When this happens, oxygen is produced. So we can measure the rate of photosynthesis. If we drew a graph, we could measure the rate of photosynthesis, like this would be our y-axis, rate of photosynthesis, dependent on how much light there is. So this could be light intensity down here. And we can measure the rate of photosynthesis by how much oxygen is produced. So rate of photosynthesis measured in O2 produced. Now the cool thing about Alodia is we can actually see the oxygen being produced in little bubbles that will come off the leaf and then float to the top. So we can simply count the bubbles. So if we increase the light intensity, we would expect to see photosynthesis increase. I'm not going to discuss if it looks more like this, or like this, or does it look like this, right? There's many different types of graphs. So we're going to talk about that later, and maybe you will even observe it in this activity we're doing. So that's photosynthesis. So remember, when we create a research question, the research question is to what extent, or you can just make like what is the effect, but to what extent does the IV affect the dV? So in this situation, our IV, our independent variable, is light intensity. So to what extent does light intensity affect our dependent variable, which will be the rate of photosynthesis measured in oxygen produced? And we will talk about how we can measure that. Okay, so that's the animation, or I'm sorry, the simulation we're going to be using. The other thing we're going to be using in this first part of this activity is just a simple Google Doc. Okay, so here I have a Google Doc here. I have my IVs, remember 5 increments. Here's my IV, so I'm measuring how many bubbles. I'm sorry, our independent variable is light intensity. So if we go back to this diagram, we can see we can set our light intensity to 100 centimeters from the plant, all the way to 200 if we wish. So we have this range that we can create. So I chose a range from 100 to 180 and increasing by 20 centimeters. You don't need to do it this way, but that's just the way I did it. So those are my 5 different increments. Also, if you remember from the module here, from what we, whoops, sorry. I'm sorry, not this one. From the, I'm lost. Where is it? Oh, this. From our little rubric that we work with, not only do we need 5 increments, we also need to have 5 repeats. So on our table here, I have 5 different trials. So as you can see here, I've already completed 4 trials because I didn't want to make you suffer through watching me do all of them. So I'm going to do the 5th one together just so you know how to do this. So I've decided that I don't want to count bubbles for a whole minute, that I'm only going to count them for 15 seconds. So I'm going to go back to our animation here. I'm going to erase this mess. Erase a little bigger so I can erase quicker. And I was working with 120 centimeters. If we go back to this, right, I'm doing the 120 centimeters from the light source, 120 centimeters from the plant. So I'm going to put the light source at 120. There we are at 120. And I'm going to start. So when you start, it automatically starts. The little timer here. So we have this opportunity to see like how many seconds have passed. The one thing I want you to change, because one of the things we need to do is process our data, which I'm not going to talk about yet. So we need to count raw data. We need to collect raw data. So I'm going to ask you to switch from this BPM button to the counter button. And all you're simply going to do is count these individual bubbles. These are oxygen bubbles being produced. Count them for a 15-second time period. And there's a little counter here, this little plus button. And all you do is you hit it every time you see a bubble. So let's start when it gets to the 9. We'll start counting at 15 seconds. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so you might have had a different number than me. I counted 35. Not 100% accurate. Or not 100% positive that's completely accurate. But you know what? It doesn't matter because we're doing multiple trials. So that is why I've done this multiple times. And if you could see, I got different numbers. So here I got 35. I'll enter that there. So what I'm going to ask you to do is to do the same thing I did, 5 trials for each, for the other, and you can use my data here if you wish, but for 100, for 140, 160, and 180, don't worry about calculating means. I just want us to simply collect the raw data.